So we looked then back at our population, knowing these data, and here I'm just illustrating that our population, or a portion of it, had 2 strains of human gamma retroviruses in that patient population from the original cohort. 1104 is the one I showed you that had the antibodies, has a full-length clone for XMRV, the second line in the database. You see it's XMRV when it's compared to the control. This is her P strain, which looks like this, but I'm sorry, the computer transformed the map a little bit bad. But this is the polytropic, and so you can see both X and P across the entire envelope region shown here in the box. So independent cloning of the viral sequences confirms the diversity in our patient population that we saw in Tony Komaroff's patient population, and the presence of actually 2 strains of virus in several of the same individuals. And this might be a clue to individuals who might be sicker if they have both as opposed to one. So we can study that. This just shows you again that the clones, when we did a lot of clones from this patient, more than 50 cultures into LNCAP, every one of them were XMRV-like, suggesting that XMRV-type virus replicates more efficiently in LNCAP. So Rachel Bagney did what's known as a phylogenetic analysis, or tree, where you compare the sequences of all the polytropic viruses in green in the gene bank, in the database, to the sequences from our patients. And I just highlight there the WPI numbers here. And you can see that many of the WPI samples from that original study also contain polytropic sequences. Interestingly, one patient contained what's called a mink cell focus, or a modified polytropic virus, much more divergent than some of the other strains. And this patient also has a different phenotype, a more aggressive disease. She's sicker, suggesting that maybe the family of viruses can be distinguished, and we might not have identified the most pathogenic gamma retrovirus first, as we did with HIV and HTLV1. Both of the one strains are the most pathogenic, but there's a lot of research to do to answer these questions on how, if and how the family members are associated with different severities of disease. So I tell you this story to answer the question that was asked earlier and explain some of our thinking about HTLV1 pathogenesis, lest you have a fear of that 20 million people getting sick. So HTLV1 causes pathogenesis. It is causative for ATL, an adult T-cell leukemia shown here, which is a very aggressive malignancy that occurs really in only 1% of the people infected with HTLV1. But HTLV1 in about 8% causes inflammatory syndromes. And one of these is known as HTLV1-associated myelopathy, or tropical spastic paraplegia. And this is a inflammatory disease where the virus infects the cells near the spinal cord and the nerves, and the patients end up in a wheelchair. Well, if you give anti-inflammatory drug, prednisone, to these patients, they'll get out of the wheelchair. Now, you don't want to give prednisone to somebody with the XMRV virus, because I showed you it's hormone responsive, and you could in fact make the situation work. But it's the inflammation that leads to the HTLV1-associated myelopathy, the neuroimmune disease, but it's actually these transactivating proteins that lead to the leukemia. So there are 2 different mechanisms of pathogenesis that cause the 2 very different diseases. So that's how we'll think about the prostate cancer and the neurological disease with the gamma retrovirus, that the mechanisms may be distinct, but the same virus can cause 2 very different diseases. So we were thinking about the inflammation signature and other ways to identify XMRV-infected patients. So we knew from Frank Persetti and Kathy Jones' work that they had found an increased inflammatory cytokine and chemokine production in the plasma from ATL patients. High levels of interleukin-6, TNF-alpha, interferon response protein 10, interleukin-8. These are inflammatory cytokines and chemokines, that could be hypothesized to be causing or leading to the disease or causing. Importantly, much lower levels in uninfected, healthy individuals. So many cytokines that should be expressed from these cells, particularly the type 1 interferons, are not expressed at all. So that's a clue also to the pathogenesis that the virus in HGRV1 has disabled the type 1 interferon response pathway. And there's a lot of nice research here. And I will show you that in our patient population of XMRV infected CFS patients, we see the exact same altered cytokine and chemokine pattern. IL-8, MIP1 alpha, beta, MCP1, IL-6, and importantly, significantly downregulated interferon alpha. Interferon alpha is necessary to stimulate the ability of NK cells and B and T cells to proliferate. So you can see we're creating now, the background or the reasons why you might not have the NK function that's been described by Nancy Klimas and others in this disease for more than 20 years. We know that MIP1 alpha is elevated in the neurodegenerative disease caused by spleen-focused forming virus. So we can begin to see patterns of inflammation that could be used diagnostically to identify XMRV infected people. So this is a summary of what we know about XMRV. While it's present in lymphocytes and APCs, it might not be the major in vivo reservoir. Infectious virus is present in both ME/CFS and cancer, and I didn't show you that Frank Rossetti has isolated XMRV from prostate cancer. We find the virus in the US. I showed you England. Meta Johnsgaard in the audience has done a nice study at VIPDX detecting XMRV and the polytropic variant in 60% of, I think, approximately 100 samples that met the criteria our patients met. And importantly, the Bonaventura Clotet group in Barcelona are also identifying infections in their patient population, so the geographic restriction or distribution is not restricted as we had hypothesized. So finally, what are the questions? I mentioned that we need to know where the virus lives. Does it live in the brain, lymph nodes, gut mucosa? Those are primary infection sites in the beginning. It's very important to the U.S. government to understand the transfusion risk given those numbers of 3.8% or perhaps 6.7% in the healthy population. We can use XMRV and the cytokine signatures I showed you for biomarkers or following or suggesting therapeutics or following for the benefit of efficacy of therapeutic strategies in a clinical trial. And of course, we're working very hard on assay development so that we can do the large epidemiological studies necessary to show that the virus is causative for any disease. Unless you do large studies across populations of the disease, you can't make those associations. So we want to know, is XMRV directly transforming? Can it cause cancer that way? Would that suggest then an increased risk or possibility of cancer in CFS populations? Can the infectious human polytropic virus be isolated? We can't call this another virus until we isolate it as we did with XMRV in the Science paper. At this point, it's just sequences. So, and does this virus play a role, alone or in combination with XMRV, in the pathogenesis of the diseases associated with it? So with that, I'd like to acknowledge my collaborators who I mentioned throughout the talk. Kei-Yong Lee and Vineet Kewal-Ramani developed that nice green fluorescent protein assay. Carrie Petro-Sadowski developed the immune serology assay that I showed you. Rachel Bagby did all of the informatics in the study. Max Post did most of the work I showed you today, but Katie Hagan and Vince Lombardi are, of course, critical to our success in other studies. All of our work is funded by the Whittemore Family Foundation. The Invest in ME group in the UK supported that UK study. The Whittemore Peterson Institute, of course, and the patients and the patient advocates. So thank you for everything, and I'll take questions.
questions? One question over there.
Yeah. Sometimes jag till little fear for the information about
Do you have a
hear me? Did you hear me? No.
Yeah,
Yeah.
I can hear you, but I didn't hear the question.
The question is, if the virus, if it's true, can you spread it further on to the family, or how?
Well, our paper showed that we could isolate virus from the blood, but we know from other work, a lot of work in the gamma retrovirus family, that you can also isolate gamma retroviruses very easily from breast milk. So the major mode of transmission of gamma retroviruses is actually breast milk. And so in HTLV as well, we cut by 40% the prevalence of HTLV in Japan and the Caribbean, and the incidence of disease by simply limiting breastfeeding to the first 6 weeks when the maternal antibodies are present, and protecting the blood supply. So, of course, the blood supply, as I mentioned, is our major focus right now. And so you can prevent these diseases by simple preventative measures. We don't know anything else about the transmission. It's important to know in the 100 years that we've been studying retroviruses, in the more than 30 years that we've been studying human retroviruses, none are contagious in the sense that they're airborne. You don't catch these through sneezing, through casual contact. Remember the early days of AIDS, when nobody would touch those people. That's not how it is here. And we know that now with HIV/AIDS, and we're certain that casual contact is not a mode of transmission. And as you all know, the epidemiology doesn't suggest that CFS is spreading that much. You know, members of your own families that never get sick and live with you your entire life. So it is not contagious in that sense. But what I show you here is work by Max Post to further answer that question, a graduate student in the lab. And he built on some work that Paul Cheney presented earlier at that Bethesda workshop. And what Paul had done is he looked at 47 people with families, actually, with disease, and found XMRV in 81% of those by the amplified culture technique. Other notable findings that I show here is 50% of all family members, not a number small at only 6 at that time, with a CFS case were XMRV positive. And importantly, various combinations of parents and offspring with XMRV had XMRV and or CFS. And one case that intrigued Max was a mother and son pair, and the mother has CFS, and the son clearly has autism. So what Max did was he looked at families from our original study shown in Science, and I just show you here the family tree. And in this particular extended family, while we didn't ever test the grandparents, the mom had the grandma had 3 different kinds of lymphoma. Both the mom and the dad, who are perfectly healthy, have evidence of infection. Dad has antibody, mom has virus that I can culture as well as antibody. There are 5 kids in the family, but only this young woman got sick, and she got sick at 12 years of age, at puberty. And again, we're thinking of a hormone-responsive virus, and we see a lot of disease start at puberty, while not necessarily the infections was there. Probably maybe her whole life, as she may have well breastfed and the mom is infected. So importantly, her cousin, the son of a CFS patient, got sick the exact same time she did, and he died of an aggressive lymphoma. So there is, the viruses, retroviruses can cause gamma retroviruses, leukemias, lymphomas, as well as prostate cancer. So we're studying the cancer rate in families with multiple family members infected. These are other families for our studies. Again, you see evidence of infection in parents, but not necessarily disease, and in siblings as well. So this sort of mimics the case with HTLV1. So if 10 to 20 million Americans are infected, maybe only 10% ever get sick. This is also true of HIV, but it's not appreciated. But Mary Carrington showed that there, by genetics, the immune response genes, that there were elite controllers. There were people who were very susceptible to replication and death by HIV and AIDS, and there were other people in the same family who were elite controllers. They had immune response gene that enabled them not to have the inflammation or the downstream effects of the virus, and we found them walking around America and continents with HIV, and they were not sick. So you don't have to get sick if you have the virus. In HIV, you can have HIV without having AIDS, but you can't have AIDS without having HIV, and it's the co-pathogens that define AIDS. When the men got Kaposi sarcoma, herpes virus-caused disease, or CMV retinitis, pneumocystis pneumonia, those are diseases and chronic infections that a healthy immune system can control. So we might expect that our hypothesis is, like HIV, XMRV causes an immune deficiency. That's not necessarily causes ME/CFS, but then a second hit, if you will, or a co-pathogen and a susceptible host with a genetic profile leads to disease in infected people.